We now invite Professor Wang Ningli, academician of the Chinese Academy of Engineering and director of the Beijing Tongren Eye Hospital. His topic is gene and cell therapy for trabecular meshwork. Welcome, Professor Wang.
First of all, I would like to take the opportunity to thank Dennis Lam gave me the opportunity to have a talk on the symposium. So I totally agree with your study. So CSF and lymphotics, that is some correlations. Finally, maybe some things we don't know related to the glaucoma. So today I will give a talk on the gene therapy for trabecular meshwork. So for the glaucoma, we know at least for the open angle glaucoma, we have two types. One is with a high pressure. Another one is normal tension. But no matter what it is, the high pressure is the target for the treatment for the high pressure glaucoma. So the trabecular meshwork is exactly the target for the treatment. So we know the glaucoma is a gene-related disease, but most of the glaucoma is the polymorphism disease, but it's not always that. Some genes like the methylins, optinurins, WT36, it is a single genes related to the glaucoma. So that it gave us the opportunity for the gene therapy. So this study is takes place in the Hong Kong with CP Pan. We work together. First, we have Chinese families. There is some new mutations. We use these Chinese families data and to do the cellular studies. This is the first time we found that these mutations can cause some kind of misfolding of the methylene proteins that can cause the ER stress and cause the apoptosis of the trabecular meshwork, cause the increase the ocular pressures. That is the high pressures glaucoma's mechanisms.
So I think for the last two talk, your talk and Clement's talk, and we have to put it to maybe the last of the.
So recently, we also repeat some study, and also we have some new findings. First, we found the different mutations also cause the ER stress. And also we found these kind of mutations can cause impairment of the deregulation on the autophages. This is something we found the new phenomenon in the glaucoma. And in our laboratory, we set up the transgenic mice for our research. We set up the different mutations from the Chinese families. There are several, at least 4 types of the mutations, the transgenic mice we have right now. So let's think about how we make the gene therapy on the trabecular meshwork to reduce the intraocular pressures. There are the 3 strategies. One is gene editing or gene silence. And secondly, we have the protein temporal, corrected the misfoldings of the proteins. And third, enhance the autophages, changes the deregulation of the autophages by editing method. So there is some possibilities for the gene therapy for the mycelium. That is some studies 50 years ago that use organ culture. There is the first time use a gene editing method to show us that it is possible the potentials reduce the intraocular pressure by this gene therapy. So recently, we use some new AVV the vectors to start over gene therapies. We use the technique silencing the mutations gene. So this study just published this year is a very fresh papers. If you like, you can check these papers. And by using this new silencing method, we got a very good result from the mice studies. We found the intraocular pressure reduced by this method, and the autoflow of the aqueous humor is increased. Finally, really, the ganglion cell, the number of the ganglion cell is the loss of the number of the loss of the ganglion cells is reduced dramatically. So, you know the AAV as vectors, still there is some things, some side effects. We don't like use that. But so we collaborated with over another teams. We created another kind of vectors. We call the virus-like particulars. This is not use the gene editing systems. We just put the protein, the Cas9 protein inside and to do the therapy. This is avoid the integration of the genes to the genomic genes. Yeah, fortunately, we did the mouse study and the monkey study. From the monkey studies, we found there are still 34% of the editing accuracy, and the safety is quite high. So this, from this result, we applied the human beings study. We use the blind eyes from the glaucoma patients. If the patients have the one blind eye, another eye is okay, still some vision. We use the blind eye as a research eye. Finally, we get approved by the our committees. We started the study. We have the two patients to get the injections of the new new vectors and with protein Cas9 proteins method. So we have just finished two patients. First one in the day 3, there are some kind of function reactions, just like the inflammation, but after several days, it becomes quiet. And the second eye, it is the same, the reaction is mild, it's not so severe. So far, the patients we follow up come to the 1 year. One patient's IOP is reduced by 20 or 30%, and the eye keeps stable, no any response, any actions. And another eyes, another patient's IOP is still keep no changes. And again, no any side effect that we found from the two individuals. So from our groups, from the first, we found the mutations, mycelium, and we found the AR stress caused by the mycelium mutations, and we set up the transgenic mice models, and we started our gene therapy by using AAV vectors and VLP vectors. So we just started our human beings study. The case is still just 2 cases. We want to increase the numbers to see if it is possible to make some changes from the bench to the bedside. So the gene therapy and the cellular therapy maybe is some future strategies for glaucoma treatment. Thank you. Thank you again.
Thank you.